Hello everyone. Today let us discuss about molecular events of fertilization. Before going to molecular events of fertilization, let us know what is fertilization. Fertilization is a result of fusion of ova and sperm which will give rise to a diploid zygote. Now let us study structure of sperm and ovum before going into a topic. Now, egg cell or the ovum is composed of many layers. First is a protective layer of follicle cells known as corona radiata. The cells will protect oocyte from any other damages. Then it is covered by a hard glycoprotein layer, zona pellucida. Then followed by a perivitelin space. Then it consists of cytoplasm, nucleus, polar body, and a packet of proteolytic enzymes known as cortical granules. This forms the structure of oocyte or an egg cell. Now let us see a sperm. Now sperm cell, first plasma membrane. At the tip of the sperm cell there is an acrosome followed by nucleus and the genetic material. Then a middle piece, this is the head region. This is the neck region, also called as middle piece, consists of mitochondria which provides energy for the motility of the sperm, then followed by an elongated tail region. This is structure of ovum and sperm. Now let's go in detail of our topic. This is molecular events of fertilization. Molecular events of fertilization is classified into several steps. First is capacitation of sperm. What is meant by capacitation of sperm? Capacitation of sperm means making a sperm ready to get fused with an egg cell. When a sperm is freshly ejaculated or freshly released, it has a poor ability to fuse with a egg cell. It has to undergo several modifications. When a sperm cell is produced, it is covered by a layer of cholesterol, and in capacitation of sperm process, it will undergo efflux of cholesterol. The cholesterol on the layer of a sperm which is deposited is removed as such. Then membrane of the plasma membrane of the sperm becomes more fragile and more permeable to a calcium ions. So influx of calcium ions can be seen. Then there will be a decrease in the pH of the sperm. Now plasma membrane is more permeable, more fragile. Okay. Then capacitation of sperm will decrease the hardenings of the sperm membrane. It will become more permeable so that it can undergo acrosomal reactions. This is all about capacitation of sperm. Now, after capacitation of sperm, it is a sperm has to go and fuse with the oocyte. For fusing with the oocyte, it has to cross several barriers. For that, first a sperm has to go through a follicle cell layer, that is corona radiata. These cells are made up of or adhered with the help of hyaluronic acid. Now sperm consists of hyaluronidase, which can make it go through the follicle cells, which can cross the barrier of follicle cells. Then binding of sperm and zona pellucida occurs. Now, zona pellucida is a glycoprotein layer. It has several proteins. Proteins of zona pellucida is basically called as ZP, zona pellucida proteins. It consists of 4 types of proteins: zona pellucida 1, ZP2, ZP3. This can also be called ZP1, ZP4. Now, ZP1 is synthesized by oocyte itself and its functionality is not that much known. ZP2 acts as a secondary receptor for the sperm. ZP3 acts as a primary receptor for the sperm, and ZP4 oocyte as such. It forms a protective barrier for oocyte. All these are proteins made up of amino acid sequence. ZP1 is of 638 amino acid sequence. ZP2 is 745 amino acid sequence. ZP3 is 424 amino acid sequence, and ZP4 is 540 amino acid sequence. Now, when a sperm cell comes and encounters with a zona pellucida, it first undergoes binding with a ZP3. And now, ZP3 glycoprotein receptor will first bind to a sperm. This is the zona pellucida, and ZP3 will act as a binding factor receptor for the sperm cell. Now, when sperm binds to a ZP3, there will be a disruption of this outer acrosomal membrane. This will disrupt and there will be a release of this acrosomal content. When an acrosomal content is released, acrosome consists of many acrosomal enzymes. Among them, acrosin is one which plays a major role in this step. Okay, and binding of zona pellucida and the sperm is characterized as a major event of fertilization, because if a sperm is binding to a zona pellucida, then only it can penetrate and get inside and the fertilization can take place, or else it is not possible as such. Now, after binding to ZP3, acrosomal content is released. Now, all the enzymes will come out. Due to disruption of this membrane, now this membrane is disrupted. So, there will be disruption of ZP3 also. ZP3 receptor will be disrupted or eradicated. Then comes a ZP2. Here ZP3, it is removed. ZP2, it acts as a secondary receptor for a sperm and the zona pellucida binding. So, inner acrosomal membrane, what is there, it will bind to a ZP2 receptor, and ZP2 receptor will help in holding sperm cell and egg in contact. Next comes the acrosomal reaction, that is what release of acrosomal enzymes and all. Then penetration of zona pellucida. Now once the sperm is binded to a ZP2, the inner acrosomal layer or membrane, now it has to penetrate inside the zona pellucida layer. Here there will be a layer called as perivitrinal space. Now the sperm has to come over here, so it has to penetrate. This is known as penetration of zona pellucida. Then after this, the process is binding of a sperm and the oocyte, or fusion of sperm and oocyte. Now, when a sperm is crossing the PVS membrane, it has to reach the cytoplasmic membrane or the plasma membrane of the oocyte. Now you remember that oocyte has a zona pellucida, PVS, and then comes the cytoplasmic layer or the plasma membrane. Now the sperm has to reach here and it has to fuse in. Now the membrane sperm head will get inside the cytoplasm of the oocyte. It will penetrate the cytoplasmic layer of the oocyte and will get inside. For this fusion, there is a protein called as CD9 which plays an important role in the fusion of the sperm and the oocyte. This process is known as sperm-oocyte binding or sperm-oocyte fusion. Then the next process is activation of the egg. Activation of egg means the continuation of meiosis. Here, we all know that in most of the mammals, when oogenesis takes place, a cell will divide into two. One is with more cytoplasmic content and other one is less cytoplasmic content, but the karyokinesis is same. This will have more cytoplasmic content and this is called as primary oocyte. And this is a polar body. Now, in case of a oocyte, before fertilization, that is before it fuses with a sperm, meiosis is arrested at the metaphase 1 stage of meiosis 1. When it fuses with a sperm, then the meiosis will take place. Again, the second meiotic division will take place. This primary oocyte will again divide into secondary oocyte or a mature ovum, and this polar body will again divide. This will be one more polar body. So total at the end of the oogenesis will get 3 polar bodies and 1 mature ovum. Okay. If this has to take place, when the sperm fuses with the egg, there must be an efflux of calcium ions from the sperm. This efflux of calcium ions will activate a cortical granules. Now you remember these cortical granules are packed with proteolytic enzymes. Now these proteolytic enzymes will reach zona pellucida and destroy at zona pellucida proteins, that is all our ZP1, ZP2, ZP3, ZP4, all will be destroyed. Why it has to destroy? Because it has to prevent polyspermy. What do you mean by polyspermy? Polyspermy is a fusion of one or more sperm, which can be a lethal to a developing embryo. Embryo wouldn't develop. Early embryonic death can be seen if polyspermy occurs. So to prevent a polyspermy, cortical granules or proteolytic enzymes of cortical granules will reach the zona pellucida and destroy the proteins of zona pellucida. Then after this comes the zona reaction. Zona reaction means hardening of the zona pellucida membrane. So far, zona pellucida membrane was permeable to sperm. So far it was permeable to sperm because it has to undergo fertilization. The cell has to undergo fertilization. Now the cause is done and no more fusion of any sperm has to take place. So the zona pellucida layer, which was permeable to sperm, will become hardened. Become more hardened so that no sperm will come and fuse with the zona pellucida. If it is trying to fuse also, it will get reverted back. No sperm can fuse with zona pellucida layer. This is known as zona reaction. After this comes the post-fertilization event. In post-fertilization event, both male and female gametes are now fused and it is enclosed within a nucleus. Now the zygote will be formed, which is diploid. Diploid zygote is formed, which is 2n in number. Then zygote will undergo a mitotic cell division. Then morula stage, followed by a blastula. Then gastrulation. Gastrulation is development of tissues layer. Gastrulation. Then implantation. Implantation is through a placenta. The mass of the cell will get attached to a uterus, wall of the uterus. Now this is all about post-fertilization event. Then the embryo will develop in the uterus as such within the coverage of amniotic fluid, and embryonic development takes place. Now let us go in summary. First is capacitation of sperm. Capacitation of sperm nothing means increasing the ability of a sperm to fuse with the ovum. Here the activity of sperm is increased. It is hyperactive. It is hypermotile now. Okay. Then binding of sperm and the zona pellucida, followed by acrosomal reactions. Acrosomal reaction is release of acrosomal enzymes, release of acrosomal content, which will release enzymes such as acrosin, which can make a zona pellucida membrane permeable through which sperm head can fuse inside. Then followed by penetration of zona pellucida. Sperm head will penetrate inside. Next, binding of or fusion of sperm and the oocyte. CD9 protein will help in the fusion of sperm and the oocyte and release of calcium ion from the sperm, and this will activate a cortical granules which will lead to a cortical reaction. Cortical reaction is movement of cortical granules proteolytic enzyme towards the zona pellucida and destroying the zona pellucida proteins. Then comes the zona reaction. Zona reaction means hardening of the zona pellucida membrane so that no other sperm fuses with it and polyspermy is avoided. No sperm can fuse, no early embryonic death can be caused due to polyspermy. Last is post-fertilization event. Development of a cell through different stages, and then finally implantation and development of a complete embryo which will give rise to a whole organism. Hope the topic is clear.